Hi, I'm Matt Spear, the Chief Medical Officer at Poseida Therapeutics. We're a cell and gene therapy company located in San Diego, California. Poseida has developed a broad portfolio of cell and gene therapy technologies, particularly transposon-based gene transfer, such as the super piggyback system, and Cas-CLOVER, a novel gene editing system. The piggyback system for gene transfer is unique in that instead of using virus to put new genes into cells, it uses a transposon, which is a plasmid that carries a cargo, i.e. the gene to be transferred into cells, that is cut out by an enzyme called a transposase and pasted into the genome to express the new gene in the target cells. This has particular advantages over viruses in the ability to carry very large numbers of genes, i.e. a large cargo capacity, and preferential favoritism for a number of cell types, such as T stem cell memory cells, or TSCM. The Cas-CLOVER gene editing system is a novel gene editing system which is similar to CRISPR and zinc fingers combined. It uses two guide RNAs to bring together the dCas9 protein along with a CLO51 endonuclease to execute the cleavage site that is targeted to be edited in the gene. This has the advantage of having a very low to no off-target editing because of the utilization of two guide RNAs. Also, it has the ability to edit resting T cells, a particular utility in creating CAR T cells. We have used this technology to create a broad portfolio of potential CAR T and gene therapy products, particularly focusing on CAR T cells at the beginning. As a reminder, CAR T cells are T cells that have been modified with the insertion of a gene for a CAR, or chimeric antigen receptor, that targets the T cells towards cancer. We have started out with autologous CAR T cells. These are T cells that are taken from an individual patient and then modified with the piggyback transposon system to carry a CAR to target these T cells to cancers such as multiple myeloma and prostate cancer. Subsequently, using gene editing system, we have been able to create allogeneic CAR T cells, where one donor can provide T cells to multiple patients. We're following this on with a number of gene therapy products to treat inborn errors of metabolism, such as OTC, or ornithine transcarbamylase deficiency. To go into a bit more detail as to what TSCM, or T stem cell memory T cells are infection or cancer-fighting cells in the body. They originate from memory cells, which may stay in the body for a long period of time, and then differentiate into effector cells, which actually do the work of killing infections such as viruses or tumor cells. So you can see why it would be useful to have a CAR T cell that originates from a TSCM and stays in the body for a long period of time. You could also imagine where utilizing TSCM T cells to create CAR T cells could be particularly useful in the treatment of solid tumors, in that as a T cell differentiates into multiple waves of effector T cells, it could continually eat into a solid tumor. As I mentioned earlier, Poseida got started creating autologous CAR T cells targeting multiple myeloma, in particular P-BCMA-101, which targets the myeloma tumor-associated antigen BCMA. Myeloma, as you know, is a common blood cancer that affects many patients in the United States every year. BCMA is an antigen expressed on almost all multiple myeloma cells. Poseida developed the CAR T cells targeting BCMA for the treatment of multiple myeloma several years ago, initiated a clinical trial in 2017, and has escalated through multiple cohorts, multiple dose levels, and was awarded an RMAT status by the FDA based off of the results of this study. This is a description of the study, a very typical first-in-man cancer study utilizing CAR T cells. The study has been conducted at a large number of academic institutions, particularly a significant number of institutions that are CIRM Alpha Stem Cell Center institutions in California. The protocol was supported in part by CIRM, and this allowed us to begin this protocol several years ago to start treating patients with this new CAR T cell therapy. The results of the clinical trial have been quite good to date. Cytokine release syndrome, which is a common toxicity associated with CAR T cells, has been relatively low, and response rates have been quite good. Poseidon next moved on to targeting PSMA for the treatment of prostate cancer with autologous CAR T cells called PPSMA-101. Like BCMA, PSMA is expressed on prostate cancer cells broadly. Prostate cancer, as you know, affects many men in the United States, and many of those patients will go on to develop metastatic castrate-resistant prostate cancer, failing all prior therapies. And thus, there's a high unmet medical need for new therapies such as this. In this case, the CAR I mentioned earlier was created to target PSMA instead of BCMA, thus prostate cancer cells. Thus far, this has been tested in mice having a model of prostate cancer called LNCAP. Quite successfully, you can see that the PPSMA-101 CAR T cells eliminated tumors in these mice on the right-hand side of this graph. Recently, we initiated a clinical trial with PPSMA-101 in patients with metastatic castrate-resistant prostate cancer. Again, the studies leading up to this trial were funded in part by CIRM, and there are a number of CIRM sites, Alpha Stem Cell Center sites, involved in the conduct of clinical trial. The next frontier in the CAR T cell space is allogeneic CAR T cells. These are CAR T cells that have had their MHC class 1 antigen and TCR removed so that one donor can create CAR T cells to treat many patients, as opposed to each donor, each patient, having to have their own T cells manufactured to treat themselves. And you could imagine where this could bring the therapy to patients much quicker with much more reproducible and consistent quality of CAR T cells. So Poseidon is using its transposon-based technology to, again, place the CARs targeting various antigens, but starting with BCMA again, into donors' T cells, but also using the Cas-Clover gene editing system to remove the TCR and MHC class 1 antigens to prevent the CAR T cells from being rejected by the patient they are transfused into, as well as eliminating the TCR to avoid a reaction against that patient's normal cells by the CAR T cells. There's another novel technology Poseidon has developed to help create these allogeneic CAR T cells called a booster technology, which allows far more doses of CAR T cells to be produced with a single donor's T cells. These CAR T cells, allogeneic CAR T cells now targeting BCMA, have been tested in a multiple myeloma preclinical model and likewise demonstrated elimination of tumors in that multiple myeloma mouse model. And we hope to bring this into a clinical trial in the near future. There are a number of other CAR T programs Poseidon is currently working on, targeting other tumor-associated antigens to treat other types of cancer. such as MUC1C, which is a unique antigen which is preferentially expressed in cancer cells of epithelial origin. So this is many common types of cancer: breast cancer, colorectal cancer, esophageal cancer, gastric cancer, and others. MUC1 is normally expressed in many cells in the body, but MUC1C, which these new CAR T cells target, is selectively expressed in tumors. And you can see below where there is significant specific anti-tumor activity in vitro for a number of these tumor types. And as you've seen in prior slides, these CAR T cells also eliminate those tumor types in mouse models, in particular here triple-negative breast cancer and ovarian cancer. The last thing I want to highlight is the ability of the transposon technology to create multi-CARs. Because of the large cargo capacity or the large number of genes a transposon can carry, we can put many different CARs into a single CAR T cell. This is an example of 4 different CARs or 4 different targets being put into a single cell, a single CAR T cell. And those CAR T cells being able to subsequently target multiple different tumor types expressing those different antigens. At Poseida, we hope to develop these into multi-targeted or bi-targeted CAR T cells, dual CAR T cells in the next several years. So overall, we've been very excited with what we have accomplished thus far with CIRM's funding and CIRM's assistance in partnership with CIRM, and look forward to upcoming potential advantages and advances in the field using this novel technology platform. Thank you.